Through a microscope. Through a microscope. Focus question. How do microscopes help people learn about the world? Barely visible. A human hair, which is about 1/10 of a millimeter, 0.0039 inches in width, is one of the smallest things that the human eye can see. Imagine, though, if you could shrink yourself until that hair looked as thick as your arm. Imagine if the hair looked as fat as a tree trunk. What would you see? The hairs on a fly's leg would look like enormous spikes. The fly itself would look like a monster with gigantic eyes. That's the close-up view a microscope provides, and it changes how we see the world. The first simple microscopes. For thousands of years, people have been interested in taking a closer look at their world. The ancient Greeks used glass globes filled with water to make objects appear larger. In the 1st century CE, the Romans discovered how to make things appear larger with glass that was thick in the middle and thin on the edges. The first magnifying glass. When people started to figure out how to make better glass lenses in Europe in the 1600s, the microscope was invented. At first, the microscope was just a sort of fancy magnifying glass. It had a single glass lens mounted above a holder for specimens. Most magnifying glasses only make objects appear about 5 to 10 times, 5 to 10 larger in size. That was the magnifying glass of the first simple microscopes. Compound microscopes. Early microscope users figured out that they could put two magnifying glasses in line with each other to get even more magnification. The second lens magnified the image from the first lens, multiplying its power of magnification. With the combination of two lenses in a single microscope, the compound microscope was born. The compound microscope's total magnification is the product of the power of both of its lenses. For example, if the first lens provides 10 times magnification and the second lens provides 40 times magnification, the specimen will be viewed at 400 times its actual size. Today, compound microscopes can magnify objects to make them appear up to 1,500 times larger than their actual size. Magnified 20 to 30 times. 20 to 30 times. One of the first early microscope users was a British scientist named Robert Hooke. Hooke improved the early compound microscopes, which could only magnify objects about 20 to 30 times their actual size. He was also one of the first people to take detailed notes on what he saw through a microscope. Hooke published his observations in 1665 in a book called Micrographia. It was the first book on microscope use written by a scientist. In Micrographia, Hooke included drawings of close-up views of a louse and a flea. He drew the compound eyes of a fly in greater detail than anyone had ever seen before. He sketched the individual hairs that cover a fly's body, the delicate antenna, and the complicated mouth parts. Hooke also sketched sections of plants and seeds. When he examined a thin section of cork under a microscope, he discovered that it was made up of hundreds of tiny chambers. He described those tiny chambers as cells. They reminded him of the small rooms called cells used by monks in a monastery. Eventually, the term cell became used to describe the tiny building blocks of all living things. Today, examining thin slices of plants lets us see how seeds are formed. It reveals how cells make up the veins and tissues of plant leaves. It shows the cells that allow water and nutrients to move in plant stems, which helps us understand how plants live and grow. Magnified 50 to 200 times. Historians believe that Hooke's Micrographia inspired another early microscope user, Antony van Leeuwenhoek. Van Leeuwenhoek was a Dutch cloth merchant, not a trained scientist. However, he used magnifying glasses to examine the different types of fabric he bought and sold. When he saw Hooke's drawings of the microscopic world, Van Leeuwenhoek started making lenses of his own. Van Leeuwenhoek discovered a way to create better lenses. As a result, he was able to make microscopes that magnified objects to more than 200 times their real size. He assembled more than 500 microscopes in the late 1600s and early 1700s. Using these stronger microscopes, Van Leeuwenhoek discovered red blood cells. He also discovered tiny life forms called microorganisms live in lake water and in the scrapings from someone's teeth. Before Van Leeuwenhoek's time, no one knew that microorganisms, living things too small to see with the naked eye, even existed. Microscopes revealed that microorganisms live everywhere. Today, if you use a microscope in the classroom, it probably magnifies objects about the same amount as Van Leeuwenhoek's early microscopes. Although 200 times magnification might not seem like a lot, it's enough to reveal details invisible to the naked eye. Even something as simple as sand is seen in a new light under a microscope. Without magnification, sand looks ordinary and plain. Under magnification, it's clear that sand from the ocean's edge contains microscopic shells, shell fragments, coral, and volcanic rock. Magnified 200 to 1500 times. Early compound microscopes had some problems because the lenses often created distortions in the image. It was extremely difficult to tell what was the actual image and what was an artificial effect caused by the way light passed through the lenses. In the early 1800s, a British scientist named Joseph Jackson Lister invented a new type of lenses that solved the dispersion problem. His work made microscopes more powerful and reliable. As a result, microscopes allowed new discoveries in multiple fields of science and medicine. In the mid-1800s, French biologist Louis Pasteur discovered that microorganisms in the air could contaminate beer or milk, causing it to spoil. Around the same time, the improved microscope allowed physicians to see microorganisms in the bodies of sick people. For the first time, scientists made the connection between microorganisms and disease. This discovery revolutionized the practice of medicine. The early compound microscope also led to the discovery that no two snowflakes are alike. American photographer Wilson Allen Bentley was known as Snowflake Bentley for his work with snowflakes. He paired a camera with his microscope in the late 1800s and early 1900s to take photographs of more than 5,000 individual snowflakes. He showed that snowflakes are mirror images of themselves in 6 different directions. Compound microscopes also allowed scientists to discover that salt, sugar, and some other substances form very specific crystal structures. Salt is made up of only 2 types of regularly spaced atoms. As a result, it forms regularly shaped cubic crystals. Sugar or sucrose is made up of 2 different, simpler types of sugar. It forms crystals that are slanted at each end and more oblong than salt crystals. The ability to see the world's tiny details can help people understand how things work. Take the cockleburr. A Swiss engineer named Georges de Mestral became curious about the cockleburrs that stuck to his clothing and his dog's fur. Wondering how they attached themselves so firmly, he took a closer look at the birds under a microscope. He saw that they had tiny hooks that latched onto loops in the fabric of his pants. Inspired by nature's hook and lip fastener, de Mestral worked to recreate the design using fabric. Years later, the result was Velcro. Velcro is made up of 2 species of fabric, one covered in thousands of tiny hooks and the other covered in thousands of tiny loops. The hooks and loops allow the 2 pieces of fabric to zip together. Magnified 1 million times. Because of the way light works, there is a limit to how much magnification a compound microscope can achieve. During the last century, scientists developed microscopes that use different ways to illuminate samples, including ultraviolet light and beams of energy called electron particles. Microscopes that use electron beams to illuminate samples are called electron microscopes. They provide the greatest magnification of any type of microscope currently in existence. They can magnify samples up to 1 million times. Conclusion. With its new degree of magnification, more detail is relieved about the tiniest structures of plants, animals, and materials. As a result, microscopes have allowed countless discoveries in nearly every field of science. Microscopes make the invisible visible, allowing us to explore the world in an entirely new way. Stains and dyes. Many cells and other small objects are transparent or see-through. This makes them difficult to see under a microscope. Stains and dyes have been used since the 1800s to make objects stand out more against their backgrounds. This makes the objects easier to see with a microscope.